Welcome to the deep dive. If you're facing down one of those foundational textbooks, you know the kind, the backbone of medicine, and you need the really high-impact knowledge delivered fast, well, you are definitely in the right place.
Yeah, absolutely.
Today, we're doing a deep dive into a fundamental mechanism, infectious disease. Specifically, we are unpacking Chapter 10 of Porth's Essentials of Pathophysiology.
And this chapter, it's really the critical link between the microbial world and, well, us, the human body.
Right.
Our mission today is to take all the intense analysis agents, virulence factors, how diseases progress, and kind of distill it down into a framework that's coherent and, importantly, clinically relevant.
Okay. So at its core, infectious disease, it's basically a conflict, right? Rooted in survival.
Exactly.
We're talking about microscopic life just trying to do its thing, survive and reproduce, and well, sometimes doing it at our expense.
Precisely. And to really get a handle on that conflict, we start with the framework the book gives us, the triad of infectious disease model.
A triad model. Okay.
Yeah, think of it like a perfect storm. You need three things. The agent, that's the microbe, then the susceptible host, the person, and finally, the environment, the conditions that kind of bring those first two together.
Oh, okay.
If you don't have all three lined up, disease doesn't necessarily happen.
Makes sense. Now, before we even get into the specific agents, the chapter lays out two really critical concepts right at the start. Colonization versus infection. What's the deal there?
Right. So colonization is basically just establishing a presence. Think about the hundreds of species of normal microflora living in your gut right now. They are colonizing you.
Okay, they're there.
They're there. They're often multiplying, but usually they're harmless. Infection, though, that's different. That's when the multiplication crosses a line and results in actual injury or damage to the host.
So injury is the key difference.
Injury is the key. Colonization is sort of a prerequisite, you could say, but it's not the same thing as infection itself.
That really sets up the whole spectrum of how we interact with microorganisms, doesn't it? It's not always warfare. Sometimes it's more like alliances.
Absolutely. The text actually defines three main types of relationships. First, there's commensalism.
Commensalism.
Yeah, the microbe benefits, gets food, shelter, whatever, but the host is completely unaffected. That describes a lot of our normal microflora.
Okay, so they benefit. We don't care.
Pretty much. Then you have mutualism, which is a true partnership. Both sides benefit.
Ah, like a win-win.
Exactly.
Yeah.
The classic example is certain intestinal bacteria. They get nutrients from us, sure, but in return, they secrete essential vitamin K. We absorb it. We need it for blood clotting. It's a good deal for everyone.
That's actually pretty cool.
It is. But of course, in pathophysiology, our main focus tends to be on the third type, the parasitic relationship.
Right. This is where things go wrong for us.
This is where the infecting organism benefits and the host sustains injury or damage. That is, by definition, the state of infectious disease.
And the severity of that disease. It's not just about how nasty the bug is it?
No, not at all. It's more of a calculation. It involves the host's health at that moment, the environmental conditions, and the microbe's own virulence.
Virulence?
Yeah.
Meaning its potential to cause disease.
Exactly. It's disease-producing potential. And this whole idea helps us define the players more clearly. You have pathogens. Those are the microbes that cause disease. And then you have harmless, free-living saprophytes, which just live off dead or decaying stuff.
Okay, pathogens bad, saprophytes generally harmless. But then there's this other category, opportunistic pathogens. That sounds tricky.
It is tricky. These are often part of our normal microflora or maybe even saprophytes that are usually benign.
Yeah.
Totally harmless most of the time.
So what changes? You mentioned host health influencing severity.
Yeah.
If they're normally okay, how does the microbe suddenly turn bad?
It's almost always about a breakdown in the host's defenses. You know, when immunity is weakened by something else, maybe malnutrition, chemotherapy, another illness. The opportunistic organism kind of senses that vulnerability. It sees its chance, seizes the opportunity to multiply rapidly, and causes disease where it normally wouldn't.
So it really highlights that the agent is only half the story.
Absolutely. The host's condition is just as critical. It's a dynamic interaction.
Okay, let's dive into the agents themselves then, the who in this whole equation. The chapter starts with maybe the strangest one on the list, prions.
Yeah, prions are.
Yeah.
They're a real biological anomaly. Discovered back in 1982, they're essentially infectious protein particles, but they lack any DNA or RNA genome. None.
Wait, no genetic material?
Yeah.
How does a protein transmit infection without a blueprint? That sounds impossible.
It sounds impossible, but it happens through this mechanism of molecular misfolding. There's a normal cellular protein called PRPC, which is stable.
Okay.
But when this normal protein comes into contact with the abnormally folded nasty form, PRPSC, the good protein instantly changes shape. It converts and misfolds into the bad form.
Like a chain reaction, a domino effect.
Exactly a domino effect, but on a molecular level. And
Yeah.
because these misfolded proteins are resistant to being broken down normally by the cell,
They just pile up.
they just accumulate. They aggregate into plaques, especially in the brain, and cause these devastating, fatal, and currently untreatable conditions like Creutzfeldt-Jakob disease or mad cow disease.
Truly terrifying stuff. Okay, moving on from protein weirdness to something slightly more familiar. Viruses. The ultimate obligate parasites.
That's a good way to put it. They're tiny. Just a nucleic acid core, either DNA or RNA. They're both wrapped in a protein coat called a capsid.
And the key thing is they have to use our cells, right?
They absolutely must hijack the host cell's machinery to replicate. They lack the equipment to do it themselves.
And the consequences of that viral takeover. This is where it gets really interesting, as the chapter points out. Sometimes the cell just bursts open, releasing floods of new viruses. Lysis.
Right, cell lysis is one outcome. But sometimes you get something much sneakier.
Latency. Latency. The virus just hides.
Exactly. It's the ultimate stealth mechanism. Think of it like the virus parks itself inside a host cell. Often, a nerve cell shuts down its active replication and just lies dormant, sometimes for years,
Waiting for
the host's immune system to be weakened or distracted.
Then.
Reactivation. The classic example is the herpes virus group. You get chickenpox, the virus goes latent in nerve cells.
And then years later, bam, shingles.
Exactly. Same virus, different manifestation because of latency and reactivation.
And we can't skip the really scary part. Oncogenic viruses. Viruses that can actually trigger cancer.
Yes, some viruses integrate their genetic material into the host cell's DNA in a way that disrupts normal cell cycle control. Human papillomaviruses, or HPV, and they're linked to cervical cancer is a prime example discussed.
Okay. Prions, viruses. Next up, the real workhorses of the microbial world. Bacteria.
Bacteria. These are prokaryotes. That means they're unicellular, they replicate on their own, but they lack an organized nucleus like our cells have.
And we classify them in a few ways, right? Shape is one.
Shape is one, yeah. Coaches are spherical, bacilli are rods, spirilla are spiral-shaped. But clinically, two other things often matter more. Their cell wall structure and their lifestyle, how they metabolize.
Let's talk cell walls. That gram-staining thing is fundamental in microlabs. Gram-positive versus gram-negative.
Okay, absolutely vital. Gram-positive bacteria have a thick layer of peptidoglycan in their cell wall. Gram-negative bacteria have a thinner peptidoglycan layer, but they also have this outer membrane.
And that outer membrane is the crucial part, isn't it? Made of lipopolysaccharide, or LPS.
Yes, that's the key insight. It's not just the chemistry of LPS, it's what happens when it breaks off. LPS acts as an endotoxin.
Endotoxin.
Because as gram-negative bacteria die and break apart, this lipid portion of the LPS gets released into the bloodstream. And that triggers a massive systemic inflammatory response.
The body overreacts.
Dramatically.
Yeah.
It leads directly to widespread blood clotting, a dangerous drop in blood pressure, basically endotoxic shock. This structural difference is probably one of the most critical clinical distinctions between the two gram types.
Wow. Okay. And their lifestyle. The chapter mentions biofilms. That sounds like something out of sci-fi.
It kind of does. Biofilms are these structured, organized communities of bacteria. They stick together, often on surfaces like prosthetic joints or catheters, and secrete this protective slime layer.
Like a shield.
Exactly. It makes them incredibly resistant to antibiotics and immune cells. And within these biofilms, they even communicate using quorum sensing.
Quorum sensing, like they take a vote?
Sort of. It's intercellular communication. They release signaling molecules, and when the concentration gets high enough, meaning there's a dense population.
A quorum.
A quorum, precisely. They collectively switch on certain genes, often virulence genes. It's like they decide, okay, team, we have the numbers, attack. This coordinated action makes biofilm infections exponentially harder to treat.
That's fascinating and slightly terrifying. The book also highlights a couple of specialized bacterial forms, Spirochetes.
Right, Spirochetes. These are bacteria with a unique helical shape and a corkscrew-like motion. Yeah. Think Treponema pallidum, which causes syphilis, or Borrelia burgdorferi, the agent of Lyme disease. That shape helps them move through tissue.
And the other one, mycoplasmas. What's special about them?
Mycoplasmas are unique because they completely lack a rigid peptidoglycan cell wall.
No cell wall. Why does it matter so much?
Because many common antibiotics, like penicillin and its relatives, work by targeting and disrupting the synthesis of that very cell wall.
Ah. So mycoplasmas are just naturally resistant to those specific drugs.
Exactly. You have to use antibiotics that target something else, like protein synthesis, to treat mycoplasma infections.
Clever little things. Okay, last group in this sort of intermediate category, the viral bacterial hybrids, Rickettsia and Chlamydiaceae.
Yeah, these are interesting. They're like viruses because they must live and replicate inside host cells. They're obligate intracellular pathogens, but they have rigid cell walls, contain both DNA and RNA, and reproduce by binary fission, just like bacteria. So they share features of both groups.
And clinically,
Rickettsia are often transmitted by arthropod vectors, ticks, lice, fleas causing diseases like Rocky Mountain spotted fever. Chlamydiaceae, especially C. trachomatis, are usually transmitted person to person and are a major cause of sexually transmitted infections.
okay, we've met the microscopic players, prions, viruses, bacteria, and these hybrids. Now we need to understand how they spread and how we track that spread. This is where epidemiology comes in, right?
Exactly. We need the language to describe disease patterns in populations. The chapter introduces two key measures: incidence and prevalence.
Incidence. That's the number of new cases, right?
New cases appearing within a specific population over a defined period of time. Prevalence, on the other hand, is the total number of active cases at any given point in time. It includes both new and existing cases.
Got it. New versus total active.
Yeah.
And then we have terms describing the geographic spread.
Right.
Endemic.
Endemic means the disease is present in particular geographic region at relatively stable expected rates. Like the common cold is endemic pretty much everywhere.
Okay. But if those rates suddenly spike,
An abrupt, unexpected increase above endemic levels in a specific area, that's an epidemic.
and if it goes global.
If the epidemic spreads across continents, driven often by modern global travel and trade, then we call it a pandemic. Think COVID-19, obviously,
Yeah.
or historical examples like the Spanish flu.
Right. So that's how we track it. Now, how does the agent actually get into the host? The modes of transmission or portal of entry?
There are several main routes. First is penetration. This means any break in the skin or mucous membranes.
Like a cut, a wound, surgery.
Exactly. Or an insect bite, an animal bite, even IV drug use, anything that disrupts that primary barrier.
Okay. Then there's direct contact.
This covers transmission through close physical contact, most notably sexually transmitted infections, STIs. But critically, it also includes vertical transmission.
Vertical. Mother to child.
Yes. Passage of infection from mother to child during pregnancy, birth, or breastfeeding. The chapter specifically calls out the torch infections.
TOR-CH. That's an acronym, right?
It is. Stands for toxoplasmosis, other, like syphilis, Zika, rubella, cytomegalovirus, and herpes simplex. These are notorious for causing serious congenital defects if transmitted vertically.
Heavy stuff. What other routes?
Well, there's ingestion, swallowing contaminated food or water.
But our stomach acid is pretty powerful, isn't it?
It is. For a pathogen to successfully infect via ingestion, it usually has to survive that extremely low pH. And this brings up the concept of infectious dose.
Meaning how much of the bug you need to swallow to get sick.
Precisely. And interestingly, factors that reduce gastric acidity, like taking antacids or certain medical conditions, can actually lower the infectious dose needed, making people more susceptible.
Huh. Didn't think about that. Last route.
Inhalation. Breathing in infectious agents suspended in droplets or dust particles.
Even though our respiratory tract has defenses like cilia and mucus.
Yes. Despite those tiered defenses, many pathogens like M. tuberculosis, which causes TB or influenza viruses, successfully invade via inhalation. It's a very effective route for them.
Okay, so the agent gets in. What happens next? The chapter describes a typical disease course with distinct stages, like a predictable progression.
Very much so. Think of it like a graph plotting the intensity of your symptoms over time. It usually follows 5 stages, though there are exceptions.
Pick one.
The incubation period. The pathogen is entered and started replicating. But you feel perfectly fine. No symptoms yet.
And this can vary hugely in length.
Massively. Could be hours for something like cholera, or literally years for HIV.
Wow. Okay, stage 2.
The prodromal stage.
Yeah.
This is when you start to feel off. Vague, non-specific symptoms start appearing. General malaise, fatigue, maybe a low-grade fever. You know you're coming down with something, but you don't know what yet.
Yeah, that feeling. Then comes stage 3.
The acute stage. This is the peak of the illness. The pathogen is proliferating rapidly, symptoms are pronounced and specific to the disease, and there's maximum impact on the body, both from the pathogen and the host's immune response.
This is when you feel the worst.
Definitely. And then hopefully comes stage 4, the convalescent period.
Things start getting better.
Yes, so the tide turns. The host's immune system is containing and eliminating the pathogen, inflammation subsides, and tissue repair begins. Symptoms start to resolve.
And finally, stage 5.
Resolution. Ideally, the pathogen is totally eliminated from the body, and there's no residual damage or lingering effects. Full recovery.
But the book mentions exceptions, right? Like fulminant illness.
Right. Fulminant disease is characterized by an abrupt, severe onset, often skipping the prodromal stage entirely. It can be rapidly progressive and life-threatening.
And you also mentioned sepsis earlier. The definition has changed.
Yes, crucially, the modern definition emphasizes that sepsis isn't just about having bacteria or toxins in the blood. It's defined as a dysregulated host response to infection that leads to life-threatening organ dysfunction.
So it's the body's reaction that becomes the primary danger.
Exactly. The immune system goes haywire, causing widespread inflammation, clotting issues, and organ damage, even if the initial infection is being controlled. It's a critical concept in modern medicine.
Okay. So we know the agents, how they get in, how the disease progresses.
Yeah.
But how do they actually cause harm? What tools do they use? This is where virulence factors come in.
Precisely. These are the specific molecules or strategies microbes use to cause disease. The text groups them nicely, starting with toxins.
Toxins, and there are 2 main types.
Yes, a critical distinction. Exotoxins versus endotoxins.
Let's start with exotoxins.
Exotoxins are generally proteins that are actively secreted by living bacteria, both gram-positive and gram-negative, during their growth.
Secreted proteins, and they're specific.
Often highly specific. They might target particular cell types or interfere with specific functions, like inhibiting protein synthesis, diphtheria toxin, or disrupting ion transport to cause massive fluid loss, cholera toxin.
And the book mentions superantigens here.
Yes, some exotoxins act as superantigens. They basically short-circuit the immune system, non-specifically activating a huge number of T cells, leading to a massive uncontrolled release of inflammatory cytokines, a cytokine storm, which can be very dangerous.
Okay, so secreted proteins, often very potent. What about endotoxins? We touched on this.
Right. Endotoxin is the lipopolysaccharide LPS component of the outer membrane of gram-negative bacteria. It's not actively secreted.
It's part of the bacterium itself.
Exactly. The danger comes when the bacteria dies and terphylize, releasing fragments of that LPS into the circulation.
And that lipid part is the toxic bit.
Yes, the lipid A portion is what triggers that massive inflammatory cascade. We talked about fever, clotting, dramatic drop in blood pressure, potentially leading to endotoxic shock. Very different mechanism from exotoxins.
Okay, toxins covered. What's the next virulence factor category?
Adhesion factors.
Yeah.
Absolutely crucial. If a microbe can't stick to host cells or tissues, it usually can't establish an infection. It just gets washed away.
So how do they stick?
They use specific molecules on their surface called ligands or adhesins that bind very specifically to complementary receptors on host cells. Think of it like Velcro, but on a molecular level.
Examples.
Bacteria might use pili or fimbriae, these hair-like appendages. Viruses like influenza use proteins like hemagglutinin to bind to respiratory cells. Some bacteria also produce a sticky slime layer or capsule that helps them adhere firmly and offers protection.
Makes sense. Can't infect if you can't connect. Third category.
Evasive factors. These are all about avoiding or neutralizing the host's immune defenses.
Immune evasion, like hiding.
Hiding is one way. Some bacteria produce thick polysaccharide capsules that make it really difficult for phagocytic immune cells, like macrophages, to engulf them. It's like they're wearing slippery armor.
What else? Fighting back.
Absolutely. Some produce leukocidins, toxins that specifically kill white blood cells. Others produce enzymes like IgA protease, which breaks down IgA antibodies, the main antibody defense found in secretions lining the respiratory and GI tracts.
So they disarm the guards. Clever.
Very clever.
Yeah.
And some pathogens, like Borrelia that causes Lyme disease, practice antigenic variation. They constantly change the molecules on their surface that the immune system recognizes.
So just when the immune system figures out how to target them?
They change their appearance, and the immune system has to start all over again. It's a constant chase.
Wow. Okay, fourth category of virulence factors.
Invasive factors. These are tools, usually enzymes, that actively help the microbe penetrate tissues and spread.
Breaking down barriers.
Exactly. Enzymes like collagenesis break down collagen in connective tissue, hyaluronidases break down hyaluronic acid, the cement between cells, and proteases degrade proteins. These basically dissolve the surrounding tissue, allowing the microbes to invade deeper.
Okay, that covers the microbes' arsenal. Let's switch gears to the clinical side. Diagnosis and treatment. How do we figure out which bug is causing the problem?
The chapter outlines 3 primary laboratory approaches. The classic one is culture.
Growing the organism in the lab?
Right. Trying to grow the suspected pathogen from a patient sample, like blood, urine, sputum, on specific nutrient media. This allows for identification and, crucially, antibiotic susceptibility testing.
But what about viruses? They don't grow on agar plates.
Correct. For obligate intracellular pathogens like viruses, you need cell cultures, growing them in living host cells in the lab. Then you look for visible signs of damage or changes to those cells caused by the virus, which is known as the cytopathic effect, or CPE.
Okay, so culture, method 2.
Serology. This is an indirect method. Instead of looking for the microbe itself, you look for evidence of the host's immune response to the microbe, specifically antibodies.
Measuring the antibody titer.
Exactly. Measuring the concentration of specific antibodies in the patient's blood. A rising titer over time suggests an active infection. This is also super important for diagnosing congenital infections.
Oh, so?
Because certain types of antibodies, like IgM, don't cross the placenta from mother to baby. So if you detect pathogen-specific IgM antibodies in a newborn's blood, it's definitive proof that the baby itself mounted an immune response, meaning it has an active congenital infection, rather than just having passive maternal antibodies, which are usually IgG.
That's a key diagnostic point. Okay, third method.
DNA and RNA sequencing, or detection, molecular methods. These are incredibly sensitive and specific.
Like PCR.
Exactly.
Yeah.
PCR, polymerase chain reaction, can amplify even tiny amounts of specific microbial DNA or RNA from a sample, allowing for rapid detection and identification. It's much faster than culture for many organisms.
And the chapter mentions real-time PCR.
Yes, real-time PCR takes it a step further. It not only detects the presence of the genetic material, but also quantifies how much is there.
Why is quantification important?
It's essential for monitoring viral load in chronic infections like HIV or hepatitis C. You can track whether the amount of virus in the patient's blood is going down in response to antiviral therapy. It tells you if the treatment is actually working.
Very powerful tool. Okay, so we've diagnosed it. Now, treatment. The chapter covers the main modalities.
Right. Broadly, we have antimicrobial agents categorized by their target. Antibacterial, antiviral, antifungal.
For antibacterials, or antibiotics, the book lists 4 basic mechanisms of action.
They generally work by 1. Interfering with bacterial cell wall synthesis, 2. Inhibiting protein synthesis, 3. Inhibiting nucleic acid synthesis, or 4. Interfering with essential metabolic pathways. Different classes of antibiotics hit different targets.
But the huge shadow hanging over antibiotics is...
Bacterial resistance, absolutely. Bacteria are masters of evolution. They rapidly develop ways to resist our drugs-producing enzymes like beta-lactamases that chew up penicillin, altering the drug's target site, pumping the drug out of the cell. It's a massive ongoing challenge.
A constant arms race. What about antifungals? Is there a specific target there?
Yes, a key difference is that fungal cell membranes contain a sterol called ergosterol, whereas human cell membranes contain cholesterol.
So drugs can target ergosterol synthesis or function?
And ideally be relatively specific for the fungus, minimizing toxicity to human cells. That's the principle behind many antifungal agents.
Makes sense. Beyond drugs.
We also use immunotherapy. This could involve giving passive immunity with intravenous immunoglobulin, IVIG, using cytokines to boost the immune response, or, of course, vaccination to prevent infection in the first place.
And sometimes you just need surgery, right?
Definitely. Surgical intervention remains crucial for source control in many infections. Draining an abscess, removing infected tissue like an appendix or an infected heart valve. Sometimes drugs alone aren't enough without removing the primary source of infection.
Okay. Lastly, the chapter touches on some bigger picture threats. Bioterrorism.
Yes, it briefly mentions the CDC categorization of potential bioterrorism agents, highlighting the Category A agents as posing the highest risk due to ease of transmission, high mortality, and potential for panic. Examples include Bacillus anthracis, anthrax, Yersinia pestis, plague, smallpox virus, botulinum toxin, and viral hemorrhagic fevers.
Scary thought. And the final point is about global spread.
Right. It emphasizes how international travel and the global marketplace facilitate the rapid spread of emerging and re-emerging infectious diseases. Think about how quickly West Nile virus spread in the U.S., or SARS, or the more recent link established between the Zika virus epidemic and the devastating outcome of congenital microcephaly.
It really underscores that infectious diseases are a constant evolving global challenge.
Absolutely. This battle between host and microbe is dynamic and ongoing.
Wow. Okay, that was an incredible density of information, but you really walked us through it. We covered the whole spectrum, really, from those bizarre prions all the way to parasites, the mechanisms of invasion like toxins and adhesion, the predictable stages of illness, and then how we diagnose and try to fight back.
It's a huge field, but Chapter 10 provides that essential foundation. Understanding these core mechanisms is key to understanding so much of medicine.
Absolutely.
And if we tie it back to the beginning, that fundamental drive of microbial evolution, it never stops. We see it most clearly, perhaps, with the rise of antibiotic-resistant bacteria. It just demands constant innovation from us.
Yep.
So thinking about all these agents and their virulence factors, their adaptability, their communication like quorum sensing, what's the single most important concept or strategy, do you think, that public health and medicine really need to prioritize right now to try and stay ahead of that evolutionary curve? Is it purely finding new drugs? Or does it require a more fundamental shift in how we think about surveillance, containment, maybe even manipulating those microbial communication systems?
That is definitely the question to chew on as you integrate this deep dive into your own studies. A huge thank you for diving deep into the often intimidating world of infectious disease mechanisms with us today.
My pleasure.
We hope this helps clarify things, and we'll catch you on the next deep dive.